What I'm going to focus on is kind of analysis of DNA sequence in two widely used applications. One is in forensics, really interesting use in solving criminal cases. And then, of course, in doing our personal DNA, we're going to talk about how this happens. So I want to begin with looking at the question of how do you actually work with DNA? So let's take this straightforward experiment that somebody might do in a lab. So you have this idea that some people are resistant to the coronavirus because they have a mutation in this spike protein. So when the virus infects you, what happens is this spike protein binds to a molecule on the surface of your cells that allows the attachment of the virus. And there are a few people who are resistant to the virus because they have an amino acid change in this receptor here, and it won't bind, and so the virus can't get into the receptor. So it'd be really useful to know exactly how the gene has changed in these people. So how would you actually go about doing that? And with modern technology, that turns out to be amazingly easy. You could do it in your garage in 2 days. So what would you do? First of all, you'd get a little sample from the person, probably just a little bit of a saliva test, and you'd want to purify some of their DNA. And the way you do that is you would take this sample, and one of the amazing things is all of this is done with really tiny amounts of material, in part because some of the reagents are expensive, et cetera, but you just don't need much. So you put it in a tiny little test tube. You'd add, about 1/2 ounce of a liquid that would dissolve all of the material in the cell. And then you'd put it in a centrifuge to get rid of everything that didn't dissolve, all the sort of particulate stuff. And you'd have left over a soup that contained tens of thousands of different molecules, if this is from human cells, that have their DNA in it, that have all their RNA, all their proteins, all their intermediates, their amino acids, their vitamins, everything would be in there. All right, you want to get out the DNA. So what you do is step one, you pour it into this little column, which you can buy. Everything done nowadays is done with kits. Builder faculty, wow, we used to do all that by hand. Nowadays you can buy a kit for everything. So this tiny little tube, it's about that big. And the key part in this tube, what is shown in that actually the red is just kind of a filter. And this material down here is simply finely ground glass. So for whatever reason, DNA, if it's at the right level of acidity and salt, will bind very tightly to a glass surface. And most other things will not. So you put your small volume from the cell extract in here, and then you put this into a centrifuge, and that makes the liquid go through there. You spin it for like 1 minute. The centrifuge is about this big. You do it right in your desktop. Put it there, pull it out, and all of that other stuff, the protein and the vitamins and the amino acids, have gone through the tube, and the DNA is stuck to this little bit of glass. So now to get the DNA off the glass, what you'd actually do is you'd then put just another liquid on top just to wash that a little bit more, another minute. And then you would put in a liquid that changes the pH and it changes the level of salt such that the DNA goes into solution and comes off of the glass. And you spin it down and you end up with your DNA, usually in a volume that's, like about 3 drops, right? Everything in the lab is done in the metric system where you say it come out in like 50 milliliters or microliters, but you're not used to using milliliters and microliters. A typical drop might be something like 50 microliters would be a drop. Okay, then you want to find out how much you got and how clean it is. And so here, actually you're going to get this in your garage. This is one thing expensive. This device probably costs a couple $1,000s. It's a spectrophotometer, but what's so cool about it is that it measures tiny, tiny amounts. just what you can barely see. What you do, you lift up this little lever here, and there's a little glass spot right there. And you put your DNA on that glass spot, and then you lower the level, and what happens is it sends a beam of light through that spot and through your sample and through your DNA, if it's there. And it analyzes what wavelength of light goes through the sample and which wavelengths of light are blocked by the sample. And so you get a spectrum like this. So this is the wavelength of light running from 230 to 350, and that is the ultraviolet range. This end would be fairly blue to the visible eye. And down here is the color of light you can't see. And if there's DNA in there, it will absorb with a maximum absorption at 280. Or 260 rather, 260. If it's protein that's been stuck in there, the peak would be here at 280. And you can measure how much DNA is by how much light was absorbed. And the machine tells you that your sample has 1,950 nanograms of DNA. Okay, so this DNA would look very clean. It's a nice clean spectrum for DNA. You know exactly how much you've got. Now, what kind of DNA is it? So, you know, is it a big piece, little piece? The next step is a frequent, highly used lab technique. Whoops. I'm getting slightly ahead of myself. Okay, so let's stick with this hypothesis. We're trying to sequence this one gene. If you actually measured how big the DNA was that you got, you would find that it is a mixture of small fragments to big fragments. Because I told you that the DNA was 6 feet long in every cell, or chopped up into 46 chromosomes. So when you break open a cell, you might think that you would break that DNA. And the answer is you do. It is very hard to break open a cell without breaking these long threads of DNA. And so they get fragmented. But in DNA terms, these fragments will often still be maybe 100,000 base pairs long. So they're broken compared to the original, but you still got big chunks of DNA, easily big enough to accommodate many genes. So it's unlikely that you're not going to break the gene, or there might be a few fragments in it, but there'd be a lot of pieces that contain your whole gene. All right, so here's the part. How do you now get the sequence of this one gene in the 20,000 that you want to find? All right, and the way you do that requires knowing a little bit about how DNA gets replicated, because making copies of the gene is essential to these techniques. So DNA replication is at some level a very complicated process. There are more than 100 different enzymes involved. But at the other level, the basic concept isn't all that complicated. So DNA is present as this double helix. You have the two strands running in the opposite direction, and they form this double helix. So step one, when DNA is to be copied, to make new copies of DNA, is there's an enzyme that pulls the strands apart and unwinds it. The enzyme that unwinds the helix is called a helicase, DNA helicase. All right. Then once the strands have been pulled apart, there is no enzyme, interestingly, that can just bind onto a single strand of DNA and copy it. All the enzymes that make DNA have to add to the end where it's double-stranded. And so what happens in the cell is there's yet another enzyme. I said there were 100 of them. There's another enzyme that puts copies the DNA to make a little piece of RNA. So this is called your primer. It's going to prime this for synthesis. So it's going to add a little RNA here. So now we've got a little place where it's double-stranded. There's a strand of DNA, there's the complementary strand of RNA over here. And the enzymes that synthesize DNA always add to the 3' end. If you looked at the chemical structure of a piece of DNA, the atoms that stick out on the 2 ends are a different arrangement. Remember, it goes sugar, phosphate, sugar, phosphate, sugar, phosphate. So those ends are distinguished, and the 3', 5', what that actually refers to is there's a sugar, 5-carbon sugar, they're numbered 1, 2, 3, 4, 5. And so one end, the 3' carbon sticks up, and the other end, the 5' carbon sticks up. But the main point is the ends are chemically distinct in their structure, and you always add to the 3' end. So what happens then is that an enzyme will come in, is recognize this as a place where it can add DNA. The nucleotides are bouncing around there, the ACs, Ts, and Gs. The enzyme takes these nucleotides, matches them up, and keeps adding them until it's copying this whole single strand here. This enzyme is called DNA polymerase, because DNA is a polymer, the polymer of DNA, and this is the enzyme that makes it. So as the 2 strands get pulled apart, the polymerase just keeps trudging along and makes a new strand of DNA. Now there's a slight complication. If it always adds to the 3' end, then down here, replication will start here and will go in this direction, will go in the other direction. But that means you've got an opening down here in which DNA can't be made until you've unspooled enough that you can add another primer. And then it'll go in that direction. So bottom line, on one strand, it's going towards the unwinding point, and the other end, you keep having to add new primers, so it's going away from this unwinding point. There's a whole set of enzymes that go in and say, oh, there's an RNA primer, I'll chew that out, we'll take that off, we'll fill that in with DNA, and so everything will be fine. In the early days of figuring this out, they observed this odd thing that there were long strands of newly made DNA, and then there were short strands of newly made DNA. And the guy who figured it out was a scientist in Japan named Okazaki. And so these are called Okazaki fragments. All right, but questions about that? Yeah, it's really they're wonderful animations that we don't have time showing this in real time, and it's really quite the whiz-bang procedure. Now, I spent some time on that because understanding how you make DNA is critical to many of the techniques that allow us to study and investigate it. And perhaps the most important of all is this, okay, is this technique called the polymerase chain reaction, PCR. How many people have heard of PCR? Okay, probably a minority of people in the room. How could you not have heard of it? What the polymerase chain reaction allows you to do is take a tiny amount of DNA, literally 1 strand of DNA, and make 1 million copies of it. Make enough that you can work with. And this is how it works. So you have your fragment of DNA. And in my hypothetical experiment, we got a little tube full of chunks of some person's DNA. Step 1 is you heat up the DNA and the strands come apart. And then step 2, to copy these, you have to put on some little bit of primer. You've got to make it double-stranded. And here's how you can fish out that 1 gene out of 20,000. The primers are an exact match to the sequence at the beginning and end of this gene. And as I said, nowadays, you can just order these. They're for $5 and they arrive overnight. So you can, it's really easy to do. So you would order a primer to one end of your gene, and you would order a primer to the other end of the gene. You make a mix of these primers, you heat the DNA, you add the primers will bind to the end. And then in your little test tube, you add the nucleotides and the enzyme that elongates this. Here we go. And it will make an exact copy of a complementary copy of this DNA, and the other primer will copy it over here. And now you started out with one strand of DNA, and now you've made two identical copies. And the only part you're copying is just the part between the two primers, which is just a tiny little piece in this 3 billion bases of DNA that's in your test tube. Then you just do that again. You heat it up, the strands come apart, the primers will bind to the single strands. You cool it down, so now that the enzyme will work and repeat it, and you repeat that cycle. And the way this really works is amazing. I mean, so what you do is you actually have a tiny little tube. For some reason, I've done these little tubes, and you put in that initial DNA that you had, just a part of a drop, and then you add in all of these primers, and you add in the enzymes to do the duplication, and you put them in a little machine about this big, a PCR machine, and what they'll do is they'll go through the heat cycles. It'll heat it up for about 5 minutes, strands will come apart. It'll cool it down to an intermediate steps, primers will bind. It'll cool it down enough that the enzymes will work. They'll duplicate it, full strand, and then repeat. It'll heat it up again. Each cycle takes about 2 minutes. And so typically you'll do that about 25 times. The whole thing takes you 2 hours, and at the end, you have replicated that gene, and you've made millions of copies of it. Pete.
Well, I'm telling you, I was in my garage, and I had a centrifuge chair, UV spectrum, I've got spectrophotometer and a UV.
Yeah.
Where I'm lost is where I go to the whole human catalog and get these reagents or enzymes. I mean, it seems like to solve the original question, you have to know the gene you want, and there has to be a catalog of hundreds of thousands of these gene things. I mean, just that's where I'm stuck. So do you blindly try 100,000 to get this gene? How do you know what gene you're looking for?
So you need to know the sequence to make your primers, is what you're saying.
Yeah, and you need to know that's the sequence that's responsible for, this particular.
Right. So in order to do this, if you're really going to fish out that gene, you need to know that sequence. And so now we have the whole genome. Okay, and so you know what protein it is. And so the answer there is a site that gives you all of that information. There are multiple ones, in fact. And this is one of anywhere in the world that you wanted to do this and wanted to look up the human genome and get that information, they would direct you to the UCSC genome browser. This is the world center for that. And so you're looking for the spike protein that causes COVID. You could literally type that in. It would pop up the sequence. You could just copy the pieces that you wanted and order those primers.
So it's fair to say that just that individual piece of information, here's the gene for the spike protein, represents like 5 years out of some researcher's life or something.
Oh, absolutely. More than 5, a lot more.
So like 100,000 cases.
Yeah. No, it's a huge community effort. Okay.
It varies.
Yes.
How long is the typical primer?
Typically about 18 bases. Yeah, so you do the math, how many do you have to have to have a unique thing with 3 billion, and turns out 18 is enough. Yeah, surprising, but still, 4 times 4 times 4, you do that sort of math, and you end up with a big number pretty quickly. Okay. All right, so we run our PCR reaction, and this method was worked out by this guy named Curie Mullis, one of the more oddball Nobel Prize winners ever. I mean, most Nobel Prizes are won by guys that spent their whole life focused on some problem that they worked on. And Curie Mullis was basically a California surfer dude. But he did work for a biotech company, and he said he actually thought of this. How can I make a whole lot of this DNA that I'm working on? Well, driving down the California coast one day, and he worked this out. And of course, there was no machine to do the heating and cooling cycles, but he had a bunch of water baths, and he tried it, and it worked. And it's kind of revolutionized molecular biology, and he won the Nobel Prize. All right. Came to Santa Cruz to give a seminar years ago. I remember. He's an interesting guy. He then became very Californian and went in his, let's just say, his own direction. All right. So if we had this DNA, we would want to see, did it work? How would we know? We know where the 2 primers are binding. We know how big the piece should be. The next procedure would be this widely used technique called gel electrophoresis. So what gel electrophoresis is, you basically have a box and you pour a thin layer of something that is very much like Jello. It's actually made out of a material called agarose. It's like Jello, gelatin. And you have a little well cut in the Jello, and you load your sample in there, and then you put an electric field across it. And the DNA will move through the gelatin layer. The small pieces will move very quickly. The big ones will keep bumping into the molecules that form the gelatin, and so they will be separated by size. Then to do something like this typically takes about an hour. You load it, you run it, takes about an hour. And then you can see DNA easily just by looking at it with ultraviolet light. We saw before that it absorbs ultraviolet light. And so you just put it on a light box with a UV source and take a picture. And wherever there is DNA, it will block the ultraviolet light in this negative that comes out as a bright band. So this would be a very typical experiment where over here you've got, you can buy little batches of DNA that are in known lengths. So this is your standard. You can judge. You can say that one there is 3,000 bases long. My gene is supposed to be 3,000 bases long. Lo and behold, the DNA that I produced from the polymerase reaction is 3,000 bases long. Large.
Doesn't the charge on the fragment affect the rate at which it goes through the cell as well as the time?
But since these are all going to be at least 100, 200, you're asking if something's really rich in GC versus AC. Well, they move different rates. Probably a little bit, but because these have many nucleotides in it, it doesn't affect us.
So it kind of averages out.
It averages out, right? And so then the last step, we want to actually get the sequence of these 3,000 bases here. And so what you would do is just literally with a little scalpel, you cut out that little piece of gel and you dissolve it and run it through another of those little DNA tubes so that you have pure DNA. And then you would put it into a little tube, you'd cap it, you'd ship it off to UC Berkeley, and for $4, can you believe this? Next morning, they give you the sequence of the 3,000 bases. So you literally could do this whole thing in your garage. All right. So whiz bang technology. All right. So how are people using this to solve some of the kind of interesting problems and analyses we want to do? And so now I'm going to get into this question, some of the applications in recent years of DNA analysis. So just to remind you that there is this interesting phenomenon in our genome that most of our DNA codes for kind of repetitive DNA. The genes themselves, the actual coding region is only about 2% of the total DNA. And then this 25% here, that's those intron regions that exist within the genes and they get cut out before the gene is read to make protein. And then there's lots of highly repeated sequences, and the other unknowns are old viruses and bits of junk DNA we picked up. So if you want to identify individuals, do you need to sequence the whole 3 billion base pairs? That would be expensive and give you a lot of information that isn't useful. So here's what they use for criminal cases. So you've heard about finding bits of DNA in blood samples or rape victims. They use semen samples, etc. And they can get just, you see a little blood stain out of the wall and they're just scratching it off and putting that in a tube. And yet there's enough DNA in there to identify, or at least you get the DNA from the person who had that blood. Well, what they use is not any of the genes, interestingly. They don't look at genes. What they actually look at is one particular class of this repetitive DNA. And it's a class called short tandem repeats, abbreviated as STRs. So what a short tandem repeat is, there are many of these scattered throughout the genomes. You'll find regions, for in this one, you have the sequence AGATA. And it gets repeated, AGATA. Actually, this one is, I read that wrong. The repeat is just 4 bases, AGAT, 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 AGAT. And in this particular one, as shown here, it's repeated 12 times. The sequence in front of it and on the other side of it, it is unique. If you looked at those 18 or 20 bases, they don't occur anyplace else in the genome. Now, the neat part about this is that we all have a version of this, but if we checked everybody in the room, what we vary is in how many times these 4 letters are repeated. So here, for example, this is just showing diagrammatically what one of these short tandem repeats, the variance that you might find in different people. All right. So it may be repeated 7 times, it may be repeated 11 times for a particular variant. There are other ones that range from 2 to 12 or 6 to 15. All right. And these short tandem repeats occur on every chromosome. So for forensic cases, they've come to an agreement, now international agreement, on which ones they're going to look at to identify the DNA of criminals, etc., or suspects. And so this is the 13 short tandem repeats used by CODIS. And CODIS is the FBI's, you see at the bottom here, the Combined DNA Index System. So here are all of our chromosomes, and on 13 of those, and there's one of them is on the Y chromosome, so you can look at this, and if you get presence of these alleles, these genes, you know it's a male. If you don't, it's a female. So they look at the short tandem repeats at 14 different places. And so the way this works out is you end up getting something that looks like a barcode. And I'm going to tell you the story of the guy who really worked this out. Alex Jeffries now known as Sir Alex Jeffries, Brett, really worked this out starting when he was 34 years old, and he's just interested in being able to identify genetic differences between individuals and not necessarily humans, but doing all sorts of evolutionary biology. And I won't go through the detail of this, but he discovered using a kind of different technology at the time that if you used a piece of radioactive DNA that was identical to these repeats, and he took it from the DNA from two different people, he would get distinct, different size DNA fragments, but they would be different in different people. And he had a flash of insight, and he said, "It looks like a barcode. Looks like a barcode that you get on a package." And he said he really actually saw it right away and thought, "This is going to have an application," you know. And so, without going into the details of how exactly they do that, what they do, if you find a little blood fragment, step one, you do that DNA purification that I showed you about. And you got a tiny amount of DNA, and you want to see the number of repeat lengths in all those different genes. So what do? You do the polymerase chain reaction. You know the primers that are at the ends of the repeat. In some people, when you make that DNA, it'll be 100 nucleotides long because it's got X number of repeats. In others, it'll be 120 nucleotides long because it's got more repeats. So you get a series of bands of different sizes. And the actual data looks like this. So it's really become high tech at this point in that any pair of primers for one chromosome might be made green, and another pair of chromosomes for the repeat, and another chromosome will be red, and another one blue. And they just put them all in one tube. and so you get a result like this. And so what you have here, in fact, is so you have, notice there's these red ones that are the same all the way across. That's just a standard that they add to it, so they know these are known molecular weights. And this is a DNA ladder here, where they can compare the sample DNAs. These are the person they're trying to identify with some standard ladder, and they can figure out how many repeats are in each of these 13 different sites. Pete.
So I'm a notorious drug lord who's left some DNA at a site. How far are we away from pulling out this garage flooding with the chromosome, CRISPRing one of these barcoded sequences and changing his genome in that respect? But it sounds like it should be like 2 years down the road from garage shop DNA.
There's a big complicated step in doing that, and that is putting that CRISPR system into your body and getting it to go to the right place, and I'll have it work. That as of today in the clinic is a $3 million office. Yeah. All right. So no, don't worry about that one yet.
Okay.
All right. So what these data look like in the end is given this example here. So these are the different genes that the CODIS system works. So it says that let's say you had a rape victim. All right. When you sample that DNA that was left at the crime scene for this particular gene, in this one it had 15 repeats and 17 repeats. Why? Because one from the mother and one from the father. So you got 2 alleles for every person. And you got 2 suspects here. And this suspect matched, but this one didn't. And that, in fact, as you go through all the different ones, you always find that pattern, 28 and 30. This suspect matched, and this one different. this suspect in fact matched every one of the results found in the evidence sample. And that says that this sample came from this person. And just in terms of how likely or unlikely this is to be right, so what this number here is says, whoops, all right, says that basically this says that 13 out of 100 people will have this kind of repeat at this gene. All right, but of those 13 people who have that, only 20% of them, in other words, I can't do my math, about 3 of them will also have the sequence at the second gene. And of those 3, only 1 out of the 3 is likely to have the sequence at the third gene. And as you keep going down, by you get to the end, it's like a 1 in 100 million chance. Now, important thing there, it does, how would it occur? Well, you're identical twins, but also if you're a sibling, then a lot of these matches are going to be the same. So it'll get you to a person or a very close relative of that person. All right. In fact, even a sibling is still going to be a 1 in a million. All right. So that's basically how that works. So it really allows you to use tiny samples and genes that for which there's global agreement. This CODIS system I was just looking it up right now has DNA from 22 million people. So how do you get into the system? You commit a crime or you're arrested for some crime. It varies state by state as to who is obliged to give a DNA sample. But so generally when they're doing in this case, for example, here they just want to, and this is the most typical one, they want to match evidence at a crime scene with a suspect. But sometimes if you just want to, you could then also put in this person's sequence here and see if they have ever been sequenced before. And if they committed some previous crime, then you've identified who the person is. The other use for this, in fact, it came up a lot in the early development, of course, is missing persons or finding people who are dead that you can't identify. So there have been lots of applications. The estimates now have shown that this has been successfully used to really solve crimes or find missing people in about 700,000 cases. So it's been a big deal. All right, I think this is a good point. Let's take about a 5-minute break, keep it short, and then we'll turn to your DNA. All right. Okay, so the new thing in DNA, of course, is getting your own DNA analyzed. And so I want to talk now about what information we get from companies that analyze our DNA and how do they do it. So here's some basic information. If we take probably almost any 2 people in this room, we'll find that our DNA is 99.9% identical. And that includes, interestingly, the repeated regions. So we got roughly 1 difference per 1,000 bases. Now, that means, though, that any 2 of us differ in 3 million different sites. And keep in mind, this would be, any 2 of us are 3 million difference, but compare ourselves to a third person, we're going to differ in 1 in 1,000 sites too, but it's not going to be just those 3 million sites that you and I differed in. There's going to be additional differences. So although we're 99.9% identical, there is a lot of sequence variation out there. The other really surprising thing is of these 3 million changes between any 2 of us, it says only 5,000 of these actually change the sequence of a protein. So you might, I got 3 million mutations, how am I surviving? Well, most of those are not in coding regions. And of those 5,000, most of those amino acid mutations do not affect the protein. After all, a typical protein will have 500 to 1,000 amino acids. If you change one of them, it's usually not in a critical part of the protein that's involved in catalyzing some chemical reaction. Most of these changes are okay. All right. So if you have this difference, a base pair difference, it's called a single nucleotide polymorphism. Polymorphic means something is different. And SNP, the acronym, and geneticists call them a SNP. So we've got, each of us has about 3 million SNPs. That's change in a single base. And when you get your DNA sequence, it means that if you sequence the whole DNA, most of the information doesn't tell you anything. It's the differences that you're looking for. So let's not sequence the whole thing. Let's just find out where the differences are. And so when you get your DNA sequence, they in fact do not sequence it. They just look for your SNPs. So how do they do it? That's what I'm going to tell you about. So right now, the companies 23andMe, Ancestry.com are probably the two big ones. They look for anywhere between 500,000 and 1 million of these single nucleotide polymorphisms. So they have reference samples from people around the world, and this is something that's growing all the time, the reference. So for Ancestry, for example, what they're trying to do, if they go to Spain, they want to get the SNP maps for as many people whose families have been in Spain a long time. After all, if that person in Spain is actually from Japan, but their family moved there a generation ago, that's not going to be an identifier for what ancestral Spanish SNPs might look like. All right, so they've got all of this reference DNA from places all around the world, and of course the total ref genome data bank grows and grows as more people get their SNPs identified. All right, so how do they do it? How do they look at 1 million SNPs? So now you cannot do this in your garage. So the big company dominating this at the moment is a company called Illumina. And they have a couple of different methods. And I'm just going to tell you a little bit about how they do this. So Illumina makes these arrays. And the key thing is, if you look at this little piece here, this little panel, this is called a microarray, which means it has little dots of DNA on this panel. And what's interesting is this can contain up to 1 million dots of DNA. So it's really high tech. So what they do is they take a silicon wafer and they etch little holes in it. And in each of these holes, they put a silicon bead. And remember, I told you how DNA sticks to glass? All right. So DNA will stick to these beads. And so step one is to take the DNA sequence next to one of these SNPs, a place where there tends to be changes in the population. And once they pick are sites that differ at least in 1% or more of the population. You can see there's a whole range. What's the likelihood that a given nucleotide is different from somebody else? And the answer is there are some nucleotides in which changes are really rare, 1 in a million. Why? Because you change that nucleotide, you die. But there are other sites that changes are frequent. So these are the somewhat more frequent changes. They also include, though, some of the sites that are known to be connected to different kinds of diseases and have physiological effects. Anyway, but to go back to this, every dot has the DNA on it that will allow them to determine one of these SNP sites. All right, so you've got your array with a little hole in it, and in that hole is this bead that contains thousands of DNA molecules. This is something I didn't emphasize enough before. With all our high technology, there are very few ways in which we can see in any way a single DNA molecule. So when you're looking at them on a gel, that band on that bright gel had millions of DNA molecules. The reason you amplify it is you got to have enough copies that you can chemically work with it, etc. And it applies to this technique too. So even though these beads are tiny, the DNA fragments are so tiny that a single bead will contain thousands of identical pieces of DNA that are associated with this one site. So what I mean by that is, let's look at here. These two pieces of DNA are identical. They're just sticking up off of this bead. And their sequence might be A, G, C, G, A. And then the next one is the place where in many people, there's a variation. And so they can identify whether your DNA, which they add to the bead now, they've chopped up your DNA in little pieces, they've added it to this collection of beads, and by hybridization, the identical sites in the test DNA and your DNA will bind to each other. And then whatever the next one up here is the SNP site. So now that they've got the DNA aligned on the chip, and the way this is done is just simply they take the chip, they put the solution in, they flood the surface of the chip. You just let it sit there for probably an hour or so until all the DNA binds. Then they add the enzymes to do DNA synthesis, and it'll add to the end of this here. So next one, they add enough to go one round of sequence. Now there's a chemical way to have it add one, but then stop. And then the next trick is that the different nucleotides, A, C, G's, and T's, the actual nucleotide is attached to something that will fluoresce with a different color. So in this example here, I'm showing these C's are green over here, and these T's are red. And so having done that, then they shine light through the chip, and they see whether it glowed green or it glowed red, for example, or whether it had some green and some red. So here's your question for today. Why would some of them look like this, the middle one? I know you've been paying attention.
You get one from your father and your mother?
Exactly. You've got two alleles for all of these. You got your mother's and you got your father's. And so then they basically, and this is where the real high tech is, you've got a million dots, but you can analyze with this sufficient resolution that you can see the color where you've got a green color for every dot on that array. In some of them, there was no green. In some of them, there was bright green, and in others, it was half green. And in that way, they can identify what nucleotide you have for every one of these 1 million dots. So they don't sequence their genes, but you end up with a catalog of your variant at all of these different sites. And then they can go back and do the analysis of these changes that we see in this region of the DNA. They might say, these exact same changes are very frequent in people who live in Portugal. And you've got them, So you've inherited some Portuguese DNA, and we know it from the collection of SNPs that you have. And the data set is so huge, 500 million, 500,000, a million, that the statistical likelihood of that becomes pretty good. So here's a kind of typical report for genetic identity, kind of tiny print for some of it, but what it will come back and it will say, all right, especially if, you know, we sequence your mother, we sequence somebody else, we found that here, the shared CN is centimorgans, the shared stretches of DNA that you shared with your mother were 3,383. And that turns out to be 50% of the total DNA that you have has the same SNPs in it that your mother has. The other 50%, you know, your father. All right. Then it might show you saying, we have a person who got their DNA sequence who had not a big piece, you know, 117 compared to 3,000, but there's 100 region of the DNA, small regions of the DNA that have exactly the same SNPs that you do. And this is equivalent to somebody who is your second cousin. All right. And so often these will show up with several sort of distant cousins. Now, how do they know that? All right. Now, there are a few people, my wife Rusty is one of them, who actually knows what his second cousin twice removed is. You tell me that. All right. So I found this neat chart here. So this is the cousin chart. It's essential to kind of interpreting these relationships. So here are you, okay, and then you have your siblings and children and grandchildren. But in terms of genealogy analysis, what's often important is your relationship, your similarity to your grandparents and your great-grandparents. So your first cousins are the children of your aunts and uncles. Your first cousins and you share the same grandparent. Your second cousins are the children of your great aunts and your great uncles, and you share a great grandparent. And so you can just work out from the fact that this person gave 50% of their DNA to this person. This one gave 50% of their DNA to this person, and keep working out down the line such that it works out. And this next chart is very busy, but it sort of shows you have your 100% of your DNA, all of your biological brothers and sisters will have 50% identical. First cousins will be 12% identical. Second cousins of the same generation will be 3% identical. And so in this way, that degree of identity gives you a pretty good idea of your relationship based on DNA sequence. Now, this has also been used for solving crimes. This is popping up more and more in the news now. So the famous example that really got this started was the Golden State Killer. So here's the story. So this was 30 years ago, and they collected DNA from the presumed killer at the crime scene. So they still had that DNA, you know, 30 years later, and now they ran it through a DNA sequence analysis program. And I'm not sure exactly what they did, but I think they were doing a SNP mapping thing like I just showed you. And what that person came out with is there were two second cousins, people with pretty similar identity. And these second cousins had people they were related to, a few of them, that had gotten their DNA sequence. And so we go down to this chart now. Here was the suspect, right? But there was a second cousin here. And there was another second cousin here. But it turned out that this, the connection here was the paternal grandmother here was the same as the paternal grandmother of this person. So they suspected that it was this person just on the percentage of DNA, but to really know, they had his DNA, they could do the CODIS type analysis, but they could not force this person to give DNA, and in fact, they didn't want the person to know that he was a suspect. So the police followed him. He went to McDonald's or something like that, had a Coke, and left it on the table. And the police came and got the DNA off the straw that he used. And it was his DNA, a perfect match. So, a whiz-bang example of how the technology is so powerful. I mean, people have done this experiment now in labs of taking an empty room and just leaving some collecting pads sitting in the room, and then having somebody come in, sit in the room for 30 minutes, and then walk out. And then they could take the DNA off the collecting pads and find out who walked in the room. Because, have you ever wondered why bloodhounds are so good, how they can go racing? Well, as you walk through here, if I walk through to that door, I will probably shed 1,000 or 10,000 cells off the surface of my head. And bloodhounds can smell that. The DNA sequencers can sequence that. And so we just live in a cloud of DNA in so many different ways. Okay. Now, there's another twist to this that I want to tell you about. And that is, I think, something that people don't understand very well, and biology students always struggle with this, was the whole fact that inheriting genetic material from your grandparents and parents, etc., has an interesting twist to it, and that is the chromosomes in your grandparents, your parents, and in you contain different collections of genes. Even though you inherited these chromosomes, they change with every generation. And how does that happen? It turns out to be very important because it really drives evolution. It increases the diversity of everybody. But the way it happens is that it's a process that occurs when any organism is making gametes. Gametes are egg cells and sperm cells. So everybody has 2 copies of every chromosome, but in the gamete, in the egg or the sperm, there's only 1 copy. And to go from 2 copies to 1 copy involves a process called meiosis. I don't know. Well, one time they had a graduation at UCSC just for the biology students. There are 3,000 biology majors at UCSC. Anyway, and so I was invited to give a little talk there. I said, what am I going to say? That I remember. So I said, nobody is graduating until they can tell me the difference between mitosis and meiosis. If you took biology, you know why that's a tough question. But what happens in meiosis is the chromosomes that you inherited from your mother and father pair. They come together and they line up side by side. And then there is a process called recombination in which they actually break and rejoin. They do this precisely, so there is no DNA sequence gained or lost at the break, but you end up with, the blue chromosome is the one you got from your mother and the red one from your father. In the egg cells or sperm cells that you're making, some of these are going to be a combination. This chromosome now is 2/3 from your mother and 1/3 from your father. And that happens at every generation. Now, 2 things. One, it means that you always get a mix of DNA, more of a mix, other than just your chromosomes mixing, the genes within each individual chromosome get mixed every generation. But that gives rise to the fact that your biological inheritance is different than your genetic inheritance. Meaning you have a great grandfather perhaps, who really was your great grandparent, father, and yet you may have none of his DNA. So how is that possible? So just let's look at how, just a single chromosome. So your grandfather has, in this chromosome we're looking at, the copy he got from his mother and his father. And the same thing with your grandmother, mother and father. But when your grandmother made egg cells, he went through meiosis, there was a recombination event such that the chromosome in her eggs was a combination of her father and her mother, which she had inherited. And then when the father makes a sperm cell, there was also a recombination between these two to produce this chromosome down here. And what has happened through recombination is that this chromosome in your grandmother down here has almost totally been lost just 2 generations later. So now, of course, you got 23 different chromosomes. So on the other ones, you're still gonna just for 2 simple generations, you're still gonna retain a lot of DNA from your great grandparent that generated this one. But you start to go back very many generations, you can get to a situation where none of that genetic material is present anymore, even though they were your direct ancestors. And of course, that complicates your kind of ancestry analysis too. So the DNA cannot go back very far before you start losing those connections. So I don't know, questions about that?
So how far, what would you say, 2 generations, 3 or 4 generations?
Probably more than that, probably getting up 5 or 6 generations, you probably start to lose any genetic trace of some, and it's random. Yeah.
Follow-up question, what is the correlation when if you go to 2 test companies like 23andMe and Ancestry, do you expect more or less the same results?
So I don't know that in much detail, but from what I've read, the answer is it's getting a lot better, simply because the data set is getting so much bigger. Yeah, early on, people were finding pretty wildly different responses from the 2, but as they keep refining it, it's getting a lot more accurate. So go ahead.
So if you know that you are the 10th generation on one of the lines of your family, there would be no way to find out genetically how you relate to that person.
Probably that far back, probably not, because you have so many great grandparents
Right,
that
so you
their
know, you
contribution.
know because of records or
Exactly,
gravestones or what have you.
that's right, right. Yeah. Pete,
So, I did 23andMe ages ago, and I knew all about the English and the Irish, the French and Swedish. But apparently I'm like a 0.3% Neanderthal, but which knowledge and stuff like that. And it was cool, I mean, I had 20 ancestors, it's fine, but, how, given this switching, how can they be sure that they had a great grandfather or a great grandmother that was Neanderthal? So many generations ago.
well, there are 2 things involved. First of all, that Neanderthal bit is scattered among all your chromosomes. Yeah, it's not just one fragment. And with different generations, you may have lost it in one place, but you inherited it from another. So it kind of all evens out in that.
Second question is, how the SNPs evolved significantly over the last, I would think that, so each of these companies is doing everything they can to maximize their profits, kind of dig the old data, tell you how to let it resort to something or something. But, it seems like the SNPs have evolved in the last year, the direction it's headed, you know that?
Well, I'm not really quite understanding what you mean by the SNPs evolved. You mean,
So if I make these plates, these semiconductor plates with the glass.
yeah,
Going after known differences. These are significant things. And over the course of 5 or 6 years, I would think the things we're looking for, how we make those plates would be different.
so I think the main difference over the years is they're adding more and more SNPs.
Okay.
Yeah, they're like making them bigger. And exactly, it speaks to this slide here. And that is the ancestry early on, 10 years ago, was pretty crude. They could say, well, you're European. All right, so like, here's the ancestry's list of, so they're saying that, you know, within Europe, they have now divided it into 1,720 specific regions. All right, so they're trying to do more fine structure, and as they get more and more data, it kind of reinforces the validity or unvalidity of that. And so a typical ancestry report, for example, will here, I'll say that in this particular person, it says 43% of your DNA, let me say this right, because this is not simple. They're using an algorithm here that's really complicated. But they would say it looks like roughly 43% of your genome came from somebody from Norway, like that. And then it goes down, 8% of your genome looks like the genomes we see in people in Sweden. So it's a real mix, but now they're getting more and more refined. Okay, and the latest thing that's really quite cool is that if your immediate relatives, especially your parents, have had their DNA tested, now they can tell you that the 43% that came from Norway came from your father. In other words, they do the ancestry per chromosome. And if they know the parent's chromosome, they can know where this ancestry originated. Okay. So let me add just two other twists to this. One is that the other thing that is used, two things that are used, is you can see your maternal line kind of unaltered by looking at your mitochondrial DNA. The important things being your mitochondrial DNA does not get significantly recombined. So the DNA that you get is in your mitochondria. You remember your mitochondria, the little organelles in your cell, and they're actually descended from bacteria that invaded the eukaryotic cells about 500 million years ago. Anyway, that DNA is separate from the DNA in your nucleus, and it doesn't undergo the same processes. And it's virtually identical in your mother and your grandmother and your great-grandmother, so it passes through unaltered. So it allows you to follow your maternal line. And the same thing can be done, okay. Oh, did I, oh, here. And for men, it's your Y chromosome. So you only have one Y chromosome. And so when you go through this making gametes, there's not another Y chromosome to pair with. So there's no recombination. And only the males get Y chromosomes. So you can use that to follow your paternal line. All right. Now, one last point. Here's my last slide for the day. So another whole part of this, and when I first thought about doing this course, I think it was partly initiated by the fact that one of Rusty's cousins had gotten her DNA done, and she'd had the health report and got these things that said you have a 23% higher likelihood of having high blood pressure, et cetera. What does all of that mean? And I know she was very confused about this, and I thought it'd be good if I could explain that. Well, I'm very confused about it. I mean, I did quite a bit of reading, and I just would not want to start to say anything about how you should interpret that sort of medical advice. Now, it raises some really interesting questions, that kind of ethics questions. For example, if somebody is, say, 15 years old, should you get their DNA sequence, and should you, get the whole medical result? I mean, there are two things, multiple things can happen. First of all, you can find out that the guy you thought was your father is not your father. All right, so this, we were in a human genetics department, we're coming to Santa Cruz, and the human geneticist said, you know, when we're constructing these family trees that will chase ancestry of diseases, this occurs more frequently than people expect. When I looked up, it's actually not that frequent. It's coming out about 1% of the. But that's still, that means 20 million people have gotten their DNA sequence. A lot of people have found out their father is not their father.
That's a big risk.
All right, then you have questions like, what if it shows that you have Huntington's? This is something, you know, like a death sentence. Do you tell them?
Yeah.
Do you ask for it? I mean, they're really interesting ethical questions. And of course, there's a whole sliding scale of this mutation is going to have this effect. This one might, this one probably not. And that's where the interpretation, and people read their report and say, oh, gee, there's a whole long list of things that I'm likely to get. And so, you know, if you do that, I think really, if you get concerned about things, you really need to talk to a medical geologist. So this is an example of something that I think this is maybe from the 23andMe side. So it just makes the point that let's have a disease called deep vein thrombosis. And what it says is between 3 and 8% of people of European descent carry one factor of this allele that makes it more likely that you'll have this disease.
Yeah.
All right, so it says 1 in 5,000 people have 2 copies. The risk for DVT for individuals in the general population is 1 in 1,000. However, if you have 1 copy, the risk increases from 1 in 1,000 to about 3 to 8 in 1,000. So I mean, so you could read something that says your chances of getting this disease are 8-fold higher. Well, it's like, oh my God, I'm doomed. No, it means instead of 1 in 1,000, it's 8 in 1,000. Still very unlikely. But, you know, should I take that seriously or not? Ask a doctor, you know, but understand what that means. Or this one down here says here's a variant in another disease-causing gene, neural tooth defect in cardiovascular diseases. But 60 to 70% of individuals in the population have one of the 2 most common gene polymorphisms, SNPs. Most of these individuals do not develop the disease. Therefore, a genetic test that identifies one of these gene variants has no real clinical implications. So my point here is that they're really subtle what these mean. And if you get the report, don't freak out, you know, and if it's really something that looks serious, go and talk to a doctor about it. All right, questions on any of this?
As you're talking about this, I wonder, is this information available to insurance companies and law enforcement?
So, I think the answer is that right now you have an opt-in, opt-out when you take the test as to how much you want your information to be available. And, you know, and that's an interesting issue too, because I mean, to me as a biologist, and I know a lot of people studying this data is so useful in really approaching some of these genetic diseases that we really want the data. On the other hand, it's people's personal privacy. So insurance companies, I'm pretty sure that legally they can't get access to them legally, but it can go into databases. Now, the other thing is that in principle, and I think this is real, I'm personally not paranoid about this. What is typically done is your DNA can go into a database without your identifier. I mean, that is what happens in a lot of these cases. Interesting what's going around globally here too. So places like Britain, there's a big movement to kind of get almost everybody's database, you know, and people are very kind of prone to participate in that. In other places, the US were more reluctant to do that.
So the more data, the better in terms of analyzing it. Are these companies proprietary applicants, are they sharing data with each other?
I think the companies are not sharing data with each other.
So one of them might have better results than the other, depending on their ability to collect data.
Exactly, yeah, and they don't, for example, tell which of the SNPs they're looking for. Yeah, so, well, that's an issue.
You know, I get a lot of emails from my heritage.com saying you may be related to this
Mhm.
person, and they're talking about forced cousins, 6 generation children. And then they show me some test results for genes, maybe the overlapping two, and then the number MCD or MD. Do you have, does that make sense what I'm saying? No, and I wonder when the sample is so small, does it even worse for my time to look at it further?
Yeah, I mean, you know, it's not a black and white answer, so it's kind of,
Cut off ways, and most likely they're not related.
yeah. Well, in terms of the relationships, you know, that is probably a little easier to interpret than the health disease thing. So, you know, if they show some relationships there, that probably means somewhere genuinely you really had some distant ancestor who had a little piece of this DNA. Yeah, okay. All right, well, thank you, everybody. So I was thinking, so what can I do to keep them coming? You know, it's complicated. So I did crime today. Next week is going to be sex.